Welcome to the Deep Dive. We're here to unpack complex topics and get you the essential insights fast.
Glad to be here.
Today, we are plunging deep into the world of bacteria. These microscopic organisms are, well, they're absolutely everywhere.
Literally everywhere. And inside us, too.
Exactly. And, that reminds me of something from when I was a kid. It kind of set up some big misconceptions for me about bacteria.
Oh, yeah. Misconceptions are often the best place to start, actually. Helps clarify things.
Totally. So, picture this. My favorite toy Stegosaurus loses its head in the bathtub.
Uh-oh. Bathtub toys. Notorious.
Right. And a few days later, my parents and I find, stuff growing inside the hollow body.
Yeah, the dreaded stuff. Probably a mix of things.
My mom told me Surgeon Stegosaurus had to be retired because bacteria had taken over. And that really stuck with me. I figured, okay, one, bacteria only grow on dirty things.
Mm-hmm. Gone blah.
And two, bacteria are always bad news. I mean, Sergeant Stegosaurus was a goner.
And that's where we jump in. Well, yeah, there were likely bacteria, maybe some mold, too, thriving in that damp toy.
Yeah.
Not exactly ideal.
Definitely not ideal.
Those two ideas. Not quite the full picture. Bacteria are, like you said, everywhere. Clean houses, outside, on your skin, absolutely vital in your gut.
So not just on dirty toys.
Nope. And always bad. Definitely not. Many are incredibly beneficial, even essential, for us and the planet.
Okay, let's really unpack this, because this deep dive is specifically for you listening, especially if you're a microbiology student aiming to become an RN.
Right.
We're pulling out the key insights about bacterial structure, function, and their clinical relevance. A lot of this ties into material covered in Dr. Remy's microbiology course at College at the Canyons.
Exactly. We want to equip you with that core knowledge you'll absolutely need for things like infection control and direct patient care.
Because this isn't just textbook stuff, is it?
Not at all. Understanding this basic microbiology, it's fundamental for making critical decisions as a nurse. Identifying pathogens quickly, figuring out why an antibiotic might or might not work, implementing the right precautions.
It bridges the micro world to the macro world of patient health.
Perfectly put. It's about connecting those dots for better patient outcomes.
So let's start with that ubiquity thing. Bacteria are everywhere, including our skin. What's the good side of that?
Well, the bacteria living on your skin, your normal skin flora, they actually form a protective layer. They kind of take up space and resources, making it harder for potentially harmful bacteria or pathogens to get established.
They're like tiny little guardians?
Pretty much. And inside you, in your gut, the microbiota is even more involved, breaking down food, synthesizing vitamins like K and some B vitamins that we can't make ourselves.
And protecting us there, too.
Absolutely. They compete with pathogens and help train our immune system. Outside our bodies, they're crucial decomposers. Nutrient cycling, nitrogen fixation for plants. Indispensable, really.
So our own bacteria, our microbiota, are a huge asset. But what happens if that balance gets messed up? This sounds important for nursing.
It's hugely important. Disrupting that balance, say with broad-spectrum antibiotics that wipe out lots of different bacteria.
The good guys and the bad guys.
Exactly. That can create an opening for opportunistic pathogens.
Right.
The classic example is Clostridioides difficile, or C. diff. It normally lives in the gut in small numbers, but if antibiotics kill off its competitors,
It can take over?
It can overgrow and cause really severe diarrhea, sometimes life-threatening colitis. It highlights just how delicate that internal ecosystem is.
So the good can become problematic if the environment changes. And speaking of problematic, let's face it, some bacteria are definitely pathogenic. What are some common infections we deal with?
Oh, there's a long list. Strep throat, tetanus, tooth decay, certain types of pneumonia, diphtheria, salmonella food poisoning, cholera.
And antibiotics are our main defense against these.
They are. But again, the double-edged sword. Broad-spectrum ones can harm our beneficial bacteria. And a critical point for anyone in healthcare, antibiotics only work against bacteria.
Right, not viruses.
Absolutely not. Colds, the flu, COVID-19, antibiotics won't touch those. That requires antiviral drugs or often just supportive care. And for prevention against both bacterial and viral diseases, vaccines are incredibly powerful.
Okay, so given these varied roles, good and bad, what are bacteria fundamentally? Let's zoom in on the basic blueprint.
All right, so bacteria are prokaryotic cells. That's a key distinction. Prokaryotic means they lack a true nucleus, the membrane-bound structure that holds DNA in our cells, eukaryotic cells.
No nucleus and generally no other complex internal compartments, right? Like mitochondria.
Correct. They don't have those membrane-bound organelles. They're typically much smaller and structurally simpler than eukaryotic cells. They're one of the three main domains of life, alongside archaea and eukarya.
But simple doesn't mean ineffective. They obviously have everything they need to live and reproduce. What are the non-negotiable parts?
Absolutely. Every bacterium has ribosomes, which are essential for making proteins. Importantly, bacterial ribosomes are 70S type, structurally different from our 80S ribosomes.
And that difference is key for antibiotics.
Bingo. It allows for selective toxicity. Many antibiotics target those 70S ribosomes, stopping bacterial protein synthesis without harming ours. They also all have cytoplasm, a cell membrane, that phospholipid bilayer controlling what gets in and out, but with hopanoids instead of cholesterol, usually.
Hopanoids, like cholesterol.
Sort of structurally analogous, yeah, they help stabilize the membrane. And of course, genetic material, usually a single circular chromosome, kind of bundled up in a region called the nucleoid. No membrane around it, just concentrated DNA.
And almost all have a cell wall.
Nearly all, yes. Outside the cell membrane is usually a rigid cell wall. It gives the bacterium its shape and, crucially, protects from osmotic lysis bursting if it takes on too much water.
Okay, those are the basics. But then there are the optional extras, the things that give certain bacteria special abilities.
Right, the bells and whistles. These are often critical for virulence, how bacteria cause disease. Things like a flagellum, a whip-like tail for movement.
Motility.
Exactly. Or a capsule, which is this extra slimy or sticky layer outside the cell wall.
What does the capsule do?
Several things. It can help bacteria stick to surfaces, like tissues in our body or medical devices. It can protect them from drying out. And importantly, it often helps them evade phagocytosis, being engulfed by our immune cells.
Makes them stealthier.
Definitely. Think Streptococcus pneumonia. The strains with capsules are much more dangerous. Then you have pili, or fimbriae, shorter, hair-like structures for attachment.
Sticking to things again.
Yes, very important for colonizing hosts. And some specialized pili are used for conjugation transferring DNA between bacteria.
We'll definitely come back to that. And plasmids.
Ah, plasmids. These are small, circular pieces of DNA, separate from the main chromosome. They're optional, but they often carry really important genes.
Like genes for...
Things like antibiotic resistance or toxins or other virulence factors, they can give bacteria a serious advantage. So understanding these structures helps you, as a future nurse, grasp how bacteria survive, cause disease, and how we can target them.
Right. That capsule making strep pneumo more virulent, or plasmids carrying resistance genes, huge clinical implications.
Yeah, absolutely.
Okay, let's talk multiplication and survival. Bacteria reproduce incredibly fast using binary fission, right? Just splitting in two.
That's the main way. Its asexual one cell divides into two daughter cells that are generally identical clones unless a mutation happens.
Yep.
And it can be very fast under optimal conditions. Some bacteria can double every 20 minutes.
Wow, that explains how infections can sometimes seem to explode overnight.
It does. It's a numbers game.
But you mentioned conjugation sharing DNA via pili. This isn't just about making clones, it's about sharing those advantageous genes, like the ones on plasmids.
Exactly. Conjugation is a major mechanism for horizontal gene transfer, spreading genes between bacteria, even sometimes between different species.
And this is a huge driver of antibiotic resistance spread.
It's critically problematic. If one bacterium develops or acquires a resistance gene on a plasmid, it can share that plasmid with its neighbors through conjugation. You can get these R plasmids that carry resistance genes to multiple different antibiotics.
Superbugs in the make-up.
Pretty much. They can spread like wildfire,
Yeah.
especially in places where bacteria are under selective pressure from antibiotics, like hospitals. There's also transformation, where bacteria can pick up free-floating DNA fragments from their environment, maybe from dead bacteria.
So understanding this genetic exchange underlines why infection control and antibiotic stewardship, using antibiotics wisely, are so vital in nursing practice.
Absolutely fundamental. We have to slow down that spread of resistance.
And if rapid multiplication and gene sharing weren't enough, some bacteria have this ultimate survival mode.
Yeah.
Endospores.
Endospores. They are truly remarkable survival structures. Certain types of gram-positive bacteria, mainly species of Bacillus and Clostridium, can form them.
When do they do that?
When conditions get really tough. Lack of nutrients, extreme temperatures, heat or cold drought, UV radiation, exposure to disinfectants. Basically, when life gets hard, they bunker down.
They form this tough, dormant structure inside the original cell.
Exactly. The original cell dies, leaving behind this incredibly resistant endospore. It contains the essential DNA and ribosomes, but it's metabolically inactive, just waiting.
Waiting for conditions to improve.
Yes. And when conditions become favorable again, the endospore can germinate back into a fully functional, reproducing bacterial cell.
The clinical significance here seems massive for healthcare settings. You said they're resistant to disinfectants.
Incredibly resistant. Many common disinfectants, even alcohol-based hand sanitizers, don't reliably kill endospores. This is why hospitals need rigorous sterilization methods, things like autoclaving, which uses high heat and pressure, or strong sporicidal chemicals like bleach.
So just using an alcohol hand rub isn't enough if you're dealing with spore formers.
Not for removing spores, no. You need mechanical friction, proper hand washing with soap and water to physically remove them. It's crucial for preventing the spread of pathogens like C. diff.
C. diff forms spores, right?
Yes, Clostridioides difficile. Its spores can persist on surfaces in hospitals for a long time, contaminating rooms and equipment. That's often how it spreads, especially after antibiotic treatment disrupts a patient's gut flora.
And other spore formers?
You have Bacillus anthracis, which causes anthrax, Clostridium tetani, causing tetanus, Clostridium botulinum, botulism, Clostridium perfringens, gas gangrene, nasty stuff. And the spores make them hard to eliminate.
Okay, that highlights the toughness. Let's circle back to the cell wall, because it's not just about protection. It's key for classification with the Gram stain. What's the core component again?
Peptidoglycan. It's this unique polymer, a mesh-like layer made of repeating sugar derivatives, N-ag and N-acetylglucosamine and N-acetylmuramic acid.
N-ag and N-M.
Right. And these chains of sugars are cross-linked by short peptide chains. It forms a strong but somewhat flexible sac around the bacterium.
Giving it shape and osmotic protection?
Precisely. And the enzyme that creates those crucial peptide cross-links is called transpeptidase. It's also known as penicillin-binding protein, or PBP.
Ah, penicillin-binding protein.
Because that's exactly what penicillin and related antibiotics, the plateletams, do. They bind to and inhibit transpeptidase.
Stopping the cross-linking.
Exactly. Without those cross-links, the cell wall becomes weak, especially in growing bacteria, and the cell eventually lyses, bursts, due to osmotic pressure. It's a beautiful example of targeting a uniquely bacterial structure.
Okay, so that's the target. Now, how does the Gram stain use differences in this wall structure? It's such a fundamental diagnostic test.
It really is. Developed way back in 1884 by Hans Christian Gram, it hinges on the difference in peptidoglycan thickness between two major groups of bacteria.
Gram positive and Gram negative.
Right. So the procedure, first you stain the heat-fixed bacteria with crystal violet. Everything turns purple.
Both types.
Both types. Then you add iodine, which acts as a mordant. It forms a large complex with crystal violet dye. This complex gets trapped within the peptidoglycan mesh.
Trapped more effectively in a thicker mesh.
That's the idea.
Yeah.
Next comes the critical step, the decolorizer, usually an alcohol or acetone alcohol mix.
What happens here?
This is where the paths diverge. In gram-negative bacteria, which have a thin peptidoglycan layer and an outer membrane.
We'll get to that outer membrane.
Right. The alcohol dissolves lipids in that outer membrane and also dehydrates the thin peptidoglycan, making it porous. The large crystal violet iodine complex gets washed out easily. They become colorless.
Okay. And Gram positives?
Gram positives have a much thicker peptidoglycan layer. The alcohol dehydrates this thick layer, shrinking the pores and trapping the crystal violet iodine complex inside. So they resist decolorization and remain purple.
Makes sense. Then the last step.
The counterstain, usually safranin, which is pink or red. The now colorless gram-negative cells pick up the safranin and appear pink or fuchsia. The gram-positive cells are already purple, and the darker purple masks the lighter pink, so they just stay purple.
Brilliant. Simple, fast, and tells you so much. If you see gram-negative Diplocaci in a spinal fluid sample,
you're immediately thinking Neisseria meningitis, bacterial meningitis, and starting appropriate treatment. It guides that crucial early empiric therapy before culture results are back.
So let's detail those wall structures. Gram-positive first. Thick peptidoglycan.
Extremely thick. We're talking 30 to 100 nanometers. Way thicker than in gram-negatives, and just a single-cell membrane inside that thick wall.
Anything else unique in the gram-positive wall?
Yes, they have teichoic acids and lipopolysaccharide acids embedded in the wall. These are negatively charged molecules that help stabilize the wall, might play a role in attachment, and can also trigger inflammation in the host. Examples. Classic examples include Staphylococcus aureus, Streptococcus pyogenes, Streptococcus pneumonia, Clostridioides difficile, and clinically, that thick peptidoglycan is the target for lactam antibiotics like penicillin and also glycopeptides like vancomycin.
Okay, now gram-negative. Thinner peptidoglycan.
Much thinner, maybe around 4 nanometers. But the big difference is they have two membranes. The inner cytoplasmic membrane, like all bacteria.
And an outer membrane.
Yes, an outer membrane outside the thin peptidoglycan layer, creating a space in between called the periplasmic space.
What's in that outer membrane? That's the key part, right?
It is. It contains proteins like porins, which act as channels allowing small molecules through. But the defining component is lipopolysaccharide, or LPS.
LPS, that sounds important.
Hugely important. Yeah. LPS has 3 parts: O antigen, core polysaccharide, and lipid A. The O antigen part varies and can be used to identify specific strains, like E. coli O157H7. But lipid A, that's the kicker. Lipid A is an endotoxin. When gram-negative bacteria die and break apart, lipid A is released. In the bloodstream, it triggers a massive inflammatory response: fever, vasodilation, drop in blood pressure, potentially leading to septic shock.
That explains why gram-negative sepsis can be so severe and rapid.
Exactly. That outer membrane also acts as a barrier. It prevents certain large molecules or hydrophobic drugs from reaching the cell.
Like vancomycin?
Precisely. Vancomycin is too large to pass through the porins in the outer membrane, which is why it's generally ineffective against gram-negative infections. You also have efflux pumps and some gram-negatives that can pump antibiotics back out.
So examples of gram-negatives.
Escherichia coli, Klebsiella pneumoniae, Pseudomonas aeruginosa, Salmonella enterica, Neisseria gonorrhoeae, Vibrio cholerae. Many major pathogens.
And briefly, there's a third wall type, acid-fast.
Yes, primarily the Mycobacterium genus, like Mycobacterium tuberculosis, the cause of TB. They have peptidoglycan, but it's linked to complex sugars and overlaid with a thick, waxy layer of mycolic acids.
Waxy layer. It sounds protective.
Very. It makes them resistant to drying out many disinfectants, and crucially, it prevents uptake of the dyes used in gram staining. You need a special acid-fast stain, like the Ziehl-Neelsen method, to visualize them.
And it makes them harder to treat.
Yes, that waxy layer is hard to penetrate. Drugs used for TB, like isoniazid, specifically target mycolic acid synthesis.
Okay, so wall structure is key. What about just basic shape and arrangement? How does that help in identification?
It's another piece of the puzzle, often combined with the gram stain result. The basic shapes or morphologies are pretty distinct. You have cocci,
Round ones.
bacilli,
Rods.
vibrios, which are curved rods like a comma. Vibrio cholerae is the classic example. Spirilla are rigid spiral shapes, and spirochaetes are flexible, corkscrew-shaped bacteria like Treponema pallidum, which causes syphilis, or Borrelia burgdorferi, which causes Lyme disease.
And how they stick together after dividing the arrangement.
Right. That depends on the plane of division and whether they stay attached. Diplo means pairs, like Diplococci neisseria, or Diplobacilli. Strepto means chains, like Streptococci, Streptococcus pyogenes, or Streptobacilli.
And Staphylo.
Irregular clusters, like grapes. That's characteristic of Staphylococcus species. You can also see tetrads, groups of 4, or Sarthinae, cubes of 8 in some cocci. Some bacilli form palisades, like a picket fence.
So gram-positive cocci in clusters immediately makes you think.
Staphylococcus. Gram-positive cocci in chains.
Streptococcus.
Gram-negative diplococci in CSF.
Neisseria meningitis.
See? Morphology and arrangement plus gram reaction give you powerful clues for initial identification and guiding that crucial early treatment.
Definitely. Let's quickly recap some other external bits and growth needs before we hit the major pathogens. Glycocalyx.
That's the general term for that sugar coat outside the cell wall. If it's well organized and firmly attached, we call it a capsule. If it's looser, more diffuse, it's a slime layer.
We mentioned capsules help evade phagocytosis and aid attachment. Slime layers, too.
Slime layers are often important for forming biofilms, communities of bacteria stuck to a surface, encased in this matrix. Biofilms make bacteria much more resistant to antibiotics and disinfectants. Think plaque on teeth or infections on catheters.
Fimbriae and pili, again, those short hairs for attachment.
Yep, crucial for sticking to host cells or surfaces. And the specialized sex pilus for conjugation.
And flagella for swimming around.
Exactly, for motility. The pattern of flagella can also help identify bacteria.
And what do they need to grow? Temperature, pH, oxygen?
Most bacteria that infect humans are mesophiles. They like moderate temperatures, basically our body temperature, around 20 to 45 degrees Celsius.
Makes sense.
They generally prefer a neutral pH around 7. Oxygen needs vary widely. Obligate aerobes need oxygen. Obligate anaerobes are killed by it. They live in places like deep tissues or the gut. Facultative anaerobes are flexible. They can grow with or without oxygen, but often prefer it.
Knowing that helps figure out where an infection might be located.
Sure. An anaerobic infection won't likely be on the skin surface.
Okay, this all sets the stage for understanding how antibiotics work. By targeting these specific bacterial features, often things our own cells don't have.
That's the principle of selective toxicity. Target the bacterial cell wall. Our cells don't have peptidoglycan. Target the 70S ribosome. Ours are 80S. Target bacterial enzymes involved in making folic acid. We get folic acid from our diet, so we don't have that pathway.
So major antibiotic classes target.
Cell wall synthesis, lactams, penicillin, cephalosporins, vancomycin. Protein synthesis, aminoglycosides, tetracyclines, targeting the 30S subunit. Macrolides, clindamycin, targeting the 50S subunit. Nucleic acid synthesis, fluoroquinolones, target DNA gyrase. Rifampin targets RNA polymerase. Metabolic pathways, sulfonamides and trimethoprim folate synthesis inhibitors, and some disrupt the cell membrane itself, like polymyxins or daptomycin.
Knowing the target helps predict effectiveness, right? Like vancomycin not working on gram-negatives because it can't get through the outer membrane.
Precisely. Or knowing that balactams primarily work on actively growing bacteria that are building new cell walls. Understanding the mechanism and target is crucial for choosing the right drug and anticipating resistance.
Okay, let's put faces to the names. Quick run-through of some key medically important bacteria nurses absolutely need to know. Gram-positives first.
Top of the list, maybe Staphylococcus aureus. Gram-positive cocci in clusters. Skin infections, abscesses, pneumonia, sepsis. And of course, MRSA, methicillin-resistant Staph aureus, a huge problem in hospitals.
Then Streptococcus species.
Right. Streptococcus pyogenes, group A strep chainzococci. Causes strep throat, scarlet fever, rheumatic fever, even necrotizing fasciitis, flesh-eating bacteria. Streptococcus pneumonia, lancet-shaped Diplococcus, often with a capsule. Major cause of bacterial pneumonia, meningitis, ear infections. Thankfully, there's a vaccine.
And the spore former, C. diff.
Clostridioides difficile, anaerobic spore-forming rod. Causes antibiotic-associated diarrhea and pseudomembranous colitis, a major healthcare-associated infection.
Okay, switching gears to gram-negatives, often associated with UTIs, GI issues, and sepsis.
Very much so. Escherichia coli, a common gut inhabitant, but certain strains cause UTIs, traveler's diarrhea, severe bloody diarrhea like a 157H7, sepsis, neonatal meningitis, a versatile pathogen.
Klebsiella and Pseudomonas, often hospital-acquired.
Yes. Klebsiella pneumonia rod with a prominent capsule causes pneumonia, UTIs, bloodstream infections, particularly in hospitalized or immunocompromised patients. Worryingly, we're seeing carbapenem-resistant strains, CRE. Pseudomonas aeruginosa, another rod, loves moist environments, causes infections in burn wounds, UTIs, lungs, especially in cystic fibrosis patients, notorious for biofilm formation and antibiotic resistance.
Others.
Salmonella enterica rod, usually foodborne, causes salmonellosis. Neisseria gonorrhea, gram-negative Diplococcus causes gonorrhea, showing increasing resistance. Vibrio cholerae curved rod causes cholera, characterized by profuse water diarrhea, rice water stools, usually from contaminated water.
In the acid-fast one.
Mycobacterium tuberculosis, acid-fast bacillus, causes tuberculosis, primarily a lung disease, but can affect other organs. Airborne transmission requires long treatment with multiple drugs due to its slow growth and resistance potential.
So knowing these guys, their gram stain, shape, typical diseases, how they spread is fundamental for nurses. Interpreting labs, anticipating problems, implementing the right precautions.
It's absolutely core knowledge for safe and effective nursing practice.
Let's tie it all together with a quick case scenario, thinking about gram-negative sepsis and also osmosis. Say you have a 68-year-old man, comes in confused, feverish, low blood pressure, hypotensive, history of a recent UTI. Blood cultures are drawn, and the preliminary gram stain shows gram-negative bacilli.
Okay, critical situation. Gram-negative bacilli in the blood of someone with a recent UPI. You're immediately thinking urosepsis, likely caused by E. coli or maybe Klebsiella.
And the connection to the cell wall.
The gram-negative status is crucial. It tells you these bacteria have that outer membrane with LPS. When they get into the bloodstream and start dying off, they release lipid A, the endotoxin,
triggering that massive inflammatory cascade,
leading to vasodilation, leaky capillaries, fluid shifting out of the blood vessels. Hence a hypotension and potentially septic shock. It's an emergency.
So nursing priorities.
Aggressive fluid resuscitation is key to restore blood pressure and tissue perfusion. And early broad-spectrum antibiotics, You also need to think about osmosis here.
Why osmosis? With the IV fluids.
Exactly. Our red blood cells don't have cell walls. If you give a fluid that's too concentrated, hypertonic, water leaves the red cells and they shrivel up crenation. If you give a fluid that's too dilute, hypotonic, water rushes in and they swell and burst hemolysis.
You need isotonic fluids.
Right. Fluids like 0.9% sodium chloride, normal saline, or lactated Ringer solution, they have roughly the same solute concentration as blood plasma, so there's no net movement of water across the red blood cell membrane. Nurses have to ensure the correct tonicity of IV fluids is used, especially when giving large volumes like in sepsis resuscitation.
And the antibiotic choice for this gram-negative sepsis?
Based on the gram stain, you need drugs active against gram-negatives. Usually, broad-spectrum lactams or carbapenems would be started empirically. But importantly, you wouldn't typically start vancomycin.
Because
Vancomycin is primarily for gram-positives. It's too large to cross the gram-negative outer membrane effectively. So knowing the gram stain result directly influences the initial antibiotic choice. The nurse's role includes monitoring vitals closely, assessing fluid status, urine output, getting those cultures sent before antibiotics if possible, and administering the right drugs promptly.
that case really pulls together the cell wall structure, the clinical impact of LPS, osmotic principles for IV therapy, and targeted antibiotic choices. It shows how fundamental this micro-knowledge is.
It absolutely does. It's not just abstract science. It's directly applicable at the bedside every single day.
Well, this has been an incredible deep dive. We've journeyed from childhood myths about dirty toys.
And decapitated stegosauruses.
Right, to understanding the intricate structures and functions of bacteria and how profoundly they impact human health and nursing practice.
And hopefully, for you listening, especially future RNs, you can see how this foundational knowledge isn't just something to memorize for an exam. It's critical thinking fuel.
Fuel for preventing infections, understanding treatments, keeping patients safe.
Exactly. It empowers you to ask the right questions, anticipate problems, and be a truly effective member of the healthcare team.
So as we wrap up, here's something to think about. Microbiology is constantly evolving. We're learning more about the microbiome. Bacterial resistance keeps changing.
It's a dynamic field for sure.
So the provocative thought for you is this. As our understanding continues to grow, what new challenges, but also what new innovations or approaches do you anticipate seeing in your own nursing practice related to microbiology in, say, the next 5 or 10 years?
How will things like microbiome therapies, phage therapy, or new diagnostic tools change how you approach infection, prevention, and treatment? It's something to keep pondering as you move forward in your career. The learning never really stops in this field.
Keep curious, keep learning. Thanks for joining us on The Deep Dive.